next session is talking about, so we always talk about clinical trials, but they really are vital in this search for a cure in whatever kind of cancer, in brain tumors. Clinical trials are the way to go, and laboratory work is needed to support moving an exciting treatment into the clinic. So our next speaker is Dr. Nader Sanai, is the J.N. Harper professor of neurological surgery at the Barrow Neurological Institute in Phoenix, Arizona. Also holds the Francis and Dion Najafi Chair in Neurosurgical Oncology. His specialty, neurosurgical practice, is devoted to patients with benign and malignant brain tumors, particularly those in high-risk brain regions. He also directs the Ivy Brain Tumor Center, a not-for-profit drug development program specializing in brain cancer drug development. If you don't know what it is, please look it up. It's really an awesome thing. Dr. Sanai is also a former ABTA grant recipient, so please put your hands together and welcome Dr. Sanai.
Well, thank you for the introduction, and what an inspiring story, and just a testament to this community we have here. I'm honored to be here. Thank you for the ABTA for inviting me. I'm honored to be here because we're all in this struggle together, and everyone who's here, as was mentioned, is here for a reason, because we're in a fight. We know that we don't have all the answers. My job in this talk, I think, is to equip you so that you can navigate yourself through all of the unknowns that you've heard a little bit about, that you will hear about. I can't tell you about every experimental therapy and every trial out there in the world, but I can equip you so that when you learn about these things, you can reach some conclusions on your own as well. So the obvious question here is, why can't we do more with what we have right now? Why haven't we found more effective treatments? You've already heard from some of the best neurosurgeons in the world. I'm a neurosurgeon as well, by the way, trained with Dr. Berger. And what we know as neurosurgeons is that the issue is not that we can't take things out safely. I think that's been shown very elegantly that we can. The issue is that that work is undone over time by the migration and proliferation of these cells and our inability to give patients and their families other therapies that prevent that from happening or reverse that from happening. So where are the new brain tumor drugs that we need to have? Well, there's a bunch of things standing in our way, some of which are self-evident that I think you all know. Number one, this is arguably the most complex cancer known to man. The slides on the left show you the normal chromosomes that most people have in their cells, 23 of them, if you remember, from grad school, undergrad, high school. On the right is what a glioblastoma set of chromosomes look like. It is extremely abnormal. So the complexity of this tumor is at another level. The second, as I think you've heard a little bit about and you probably are aware of as well, is that drugs come from the pharmaceutical biotech industries, and they are inherently trying to do good work, but they are driven by market forces, and market forces drive their innovations to other cancers that have larger market sizes, and that's just a reality we have to face. It's not to demonize them by any means at all. It is something that we have to navigate, but you can very easily predict where new drugs are coming from just by the size of the market that those indications serve. And this is borne out in the actual survivorship in the United States and worldwide. Cancers that really are within large market sizes have had the greatest gains in survivorship, whereas brain cancers and CNS cancers have not. I think you also heard earlier about where the federal support for this kind of effort starts and finishes. The National Cancer Institute is the federal organization largely charged with helping us get new drugs. And its budget has grown, which is great, but proportionally, its allocation to CNS cancers has diminished. And that's a big problem. And that's been evident to those of us in the community for many years. 2 decades ago, there were many different organizations funded by the federal government to try to find new drugs to test and test them at clinical trial levels. And over the years, those consortia have diminished in size and in scale. And just this 2-3 years ago, the last of these consortia, the ABTC, was defunded. So this is where an organization like the ABTA really shines, because it gives us a bridge to these goals that otherwise we're not seeing a lot of support for. It's also important to understand that the mechanisms through which new drugs develop are really within a capitalist environment of biotechs and biopharma. And biotechs and biopharma don't always take a direct line to getting us the new drugs we want. Sometimes it's a zigzag. Sometimes the incentivizations for the biotech models can decouple from what's actually the clinical results. So you can have drugs or therapies where the clinical results are not that spectacular, but yet they continue to be developed because there are other business arguments to be made for doing that. And that can create a little bit of scatter within our space in terms of where all of our efforts and our patients go. And finally, I think something that Dr. Miller pointed out in an editorial several years ago in JAMA, which is one of the medical association journals, is that the community of scientists and surgeons that you all have heard from today and over the years have met, these are high-achieving people, and it can be difficult to pivot away from a concept that you've dedicated your whole life to. It can be difficult to recognize that a new strategy actually is not promising, and we need to shut it down and move the funding elsewhere and move our attentions elsewhere. So this can be also a challenge for us as a field. That being said, clinical research is clearly the way for us to get better results, and there is example after example of that across the country and world. Melanoma, as you know, used to be a death sentence if it was metastatic, and today for many patients it can be survivable. That is simply because of the rise of what's called checkpoint inhibitors and immunotherapies that were developed and have clearly worked in improving survival. So clinical research can get us there. Basic science research gives us the foundation for that. It's up to us to put it together. And so what I'm going to do in the next few minutes is socialize you to the language that you're going to hear, you've heard from us today, you're going to hear and read about in the months and weeks to come. You understand research, you've heard this term translational research, which is really research that translates or brings a scientific concept to the patient. On the other end of that spectrum, there's something called discovery-based research, where you're really trying to understand fundamental mechanisms of biology. And it's a spectrum, as you can see here, where you go from the left to the right, getting you closer and closer to applicable avenues that give you new drugs. And that spectrum has to be supported by something. And you can see here that the spectrum of support ranges from federal funding to private and industry funding to everything in between, including foundations like the ABTA. And these are big numbers. These are big numbers because these are costly studies that are pretty technologically advanced, and in the cases of clinical trials, are extremely high on scale and complexity. But it all starts in the laboratory, and many of the most essential concepts in oncology care today arose from really basic fundamental discoveries in the laboratory. And this is where our system can get a little bit wobbly in the sense that all of the premises for drugs today originated from typically an animal model in the laboratory, giving us an idea of what might work in this animal, might work in patients. But we've seen over and over in the brain tumor community that what works in animals hasn't always worked in patients. We've seen that with checkpoint inhibitors, the same drugs that can be almost curative for some melanomas. We've seen that with vaccine therapies, a strategy that can work in other cancers, but so far in brain tumors hasn't borne out, even though in animals it seems to be effective. We've seen that with what's called precision medicine. Precision medicine is when you take the genetics of a patient's tumor, as you probably know, you match it to a drug and you try to exploit that gene target. And that strategy can work in the laboratory, but so far in major randomized phase 3 trials, it has not. We've seen that in what's called antibody-drug conjugates. You may have heard of a drug called Depatux, which is an antibody that homes into a tumor and is linked to a poison. And so you give it to the patient, it homes into their tumor, and in an animal, it has a pretty dramatic effect, and in a very large phase 3 study in glioblastoma patients, it did not. And we've seen that in other combinations of drugs. So most recently, many of us in the field were at a meeting where we heard about a chemosensitization phase 3, combining Temodar and another drug what's called a PARP inhibitor together. This was a strategy that had a lot of evidence for it in the laboratory and inspired us to invest years of effort, many patients volunteering themselves for it, and obviously a lot of money, and ultimately the study was negative. So as Dr. Berger alluded to, and others have as well, there is another way to add some texture to this discovery process, and that is to ask the same kinds of questions we ask in the laboratory in ourselves, in our patients. And these are a series of types of trial designs. One version is called window of opportunity. One version is called the phase 0 trial. The basics of it are this. In a phase 0 trial, you're a patient, you're going to the operating room anyways. We give you a small dose of drug just to see how that drug performs in your tumor. That's the end of the study. You don't get anything afterwards. It is simply a way to test whether a drug is performing as expected in humans. A window of opportunity is when there's something called therapeutic intent, meaning you're already getting some experimental therapy. You've been getting it for days or weeks, and then you're going to the operating room. So we've had it in your system for a while. Now we're going to see how your tumor has reacted to it, and then you're going to continue to get it afterwards. And at my program at the Ivy Center, we do something called PKPD triggered studies. These are trigger studies because you get a drug that's been selected for whatever tumor you might have. You go to the operating room, and we test to see if that drug is working, but only if it's proving to us that it's doing something do you continue to get it afterwards. If not, then you're off the study and you go on to something else without losing any time. These kinds of studies are all predicated on basic research, and basic research has two general forms that you may be hearing about. What we call hypothesis testing, meaning I have an idea of how to exploit glioblastoma, and I'm going to write out this idea of how this mechanism might work, and I'm going to test it specifically in the laboratory or in a patient or in a clinical trial. And then there's hypothesis generating. This means I don't really have a complete idea of how something may work, but I'm going to gather a lot of data. Maybe I'm going to take 1,000 patients' tumor samples and just study it across the board and find some pattern. And that pattern is then going to become the basis of a hypothesis. So it's important for you guys to understand this because, number one, these are the kinds of studies that we support in our community, and it's good to know what you may be part of or what you may be supporting, but also to know that there has to be a balance between these two things. Especially for those of us in this room, we want to get to the point, which is how do we get new agents, new drugs, developed, tested, and proven? When you get to the clinical trial stage, that's when you talk about things called investigator-initiated versus industry-sponsored trials. So this comes down to who's in charge of the trial that I'm on? Who conceived of it? Who's paying for it? In an investigator-initiated trial, it's the investigator that has an idea. That investigator might be a scientist or professor or a surgeon or a doctor. Then they go get funding for it, typically from the ABTA, the federal government, NIH, NCI. They get access to some new therapy that is part of their hypothesis for how this could be working in patients. And then they go to an academic medical center typically, and they do that trial with that experimental therapy. So in these trials, it's really being run by the investigator. There's another version of this, which is also very common, and one is not worse than the other, and that's when you have a company, maybe a biopharma, biotech, and they have a new therapy, and they need funding, so they secure it from themselves or their investors, and they hire what's called a CRO, an organization that does clinical trials for a living, and they pay that organization to then go to academic medical centers and pay them to do these trials for them, in addition to community sites. The reason it's important to understand this distinction is because this is how we determine who makes the decisions about how these trials are done. If it's the first category, well, then most of those decisions were really made by the investigational team. If it's the second category, well, then it's really a partnership between the investigational team and the biotech that is heavily invested in this. So if we put these together, how does this help us inform the therapies that we've been hearing about today, online, from our doctors and physicians, things that are on the horizon? So I mentioned precision medicine. Precision medicine is this concept that we're all different, and all of our tumors are different. and the biology of our tumors are different. And so therefore, the treatments for these tumors should be tailored to those individual biologies. I'll give you an example. BRAF V600 is a mutation that can be found in some tumors, some brain tumors and other non-tumors. And we understand the mechanisms around this mutation. And in fact, there are 2 drugs recently developed, the ones on the right, that target 2 things related to this mechanism. And so there was recently a type of clinical trial called a basket trial. A basket trial is when we don't care what kind of tumor you have. You could have a brain tumor, a lung tumor, a kidney tumor. What we care about is whether it has this specific mutation. And if it has this mutation, then you're in. And we're going to give you these 2 drugs. And what you see in these vertical bars are patients with different kinds of tumors. So over on the left, these are patients with high-grade gliomas, low-grade gliomas that are adults. Over on the right, these are pediatric glioma patients. And in the middle, you have everything from lung cancer to breast. And what you see in the percentage is the percent of patients that are responding to the drug. So what this told us is that there was a population of kids with low-grade gliomas with this mutation that seemed to respond. And you see the imaging on the bottom, the big blotchy white thing in the center of this kid's head disappearing. This is a response to this drug because of this precision medicine approach. And this is why this drug, just a few months ago, this combination of drugs got approved for exactly this indication. So this is how taking some of these principles into practice can give us new drugs, especially if you're a father or mother of a child with something like this. What about immunotherapy? We've heard a lot about this today, and there are different versions of it. There are checkpoint inhibitors that kind of take the brakes off your immune system. There are cancer vaccines that prime your immune system. There are CAR T cells where you're injecting immune cells into your body that are supposed to home in to your cancer. And there are antibody treatments, etc. These are all in development. Maybe the one that you've heard about perhaps the most is DC-Vax. It's been in the news. It's had a major trial surrounding it. DC-Vax is a vaccine, a cancer vaccine made by a biotech, where they take tumor cells from patients, process them, prime immune cells against them, and then inject those immune cells back into you. And there was a major phase 3 clinical trial where 331 patients were randomized, some of them to the vaccine, some of them to placebo. And the challenge in this trial was that a lot of the patients that got placebo ended up having some recurrence, and they crossed over, meaning they said, okay, this didn't work for me, I got placebo, now I'd like the vaccine. And they got the vaccine and went into the other group. And the reason that's problematic to interpret is because sometimes, as you saw from Tumor Board, you can see things on a scan where you say, aha, the tumor's recurring, like this scan on the left, but then if you gave it enough time, it would go away because it was pseudo-progression. So sometimes when we think something's recurring, it's actually not. And this was a challenge in this trial because all of their placebo group kind of migrated away. And in the end, they were left with a large group of patients that got the vaccine and not an easy way to compare them. So what they did was they took a population of other studies from other clinical trials, and they said, look, this is how good those trials did, and this is how good our drug did. And so you see the DC-Vax curve. By the way, this is a survival curve. I think you've been shown this, but maybe no one's talked you through it. Survival curves are very simple. The vertical bar is the percentage of patients that are alive. The horizontal bar are the number of months it's taken since the intervention. So as the curve goes down, that's people not surviving this treatment and not surviving that disease. And so the higher the curve relative to one another, the more improvement you're seeing. And so you see here, there's a little bit of separation in the curves, but of course the challenge is, are we really comparing apples to apples? This is going to be something you're going to watch unfold in the months to come. Will this drug get approved by the FDA based on this kind of data? We don't know. You may hear controversy around it, and this is why I'm trying to explain to you why there's going to be some controversy around it. What about viral therapy? I think we already heard a little bit about the polio virus therapy. In fact, there are many different types of viral therapies in development. We've heard a lot about some, but in fact, these are all of the ones that are currently in clinical trials somewhere in the US. The one I've circled is the one that you've heard about the most, which is the poliovirus study that originated at Duke. And as was described, this is a study where You have an operation, and in that operation, you have a catheter placed, and during that operation, that catheter infuses the poliovirus for a long period of time, and then you go on to recover from that. And this is the data from the New England Journal paper that was alluded to earlier, and these are the survival curves. And when people talk about the tail, Dr. Miller talked about the tail of a survival curve. What she's alluding to is that, see how that blue line never gets to the bottom? Because there's a small population of patients here that did much better than everyone else. And so the question when you look at this data is that because of the poliovirus therapy, or were these just patients that were really going to do well no matter what? That's really hard for us to sort out. In this trial, the comparison was, again, not a group of patients that were randomized, but just a group of patients that happened to be treated around the same time that had similar characteristics. So is this enough to conclude that this therapy is effective? We don't know. This is why there's more trials being done around this. And here's one of their best examples. This is the patient that lived, or at least was at 58 months out. You see this right frontal tumor and the reaction to the poliovirus, and then the subsequent resolution of it over many, many months. We heard from Dr. Yu about radiation. Radiation is arguably one of the most effective therapies we have. It's also the most ubiquitous therapy we have. In the United States, you can get chemotherapy, surgery, radiation. Across the world, though, those things aren't always available, but radiation is probably more prevalent than anything else. It's a key element for us to treat these tumors, even though we don't like the side effects when it happens. So trying to accentuate radiation effects is important, and all the things are circled are little vulnerabilities within a tumor cell's response to radiation that can be targeted. So here's an example of a phase 0 clinical trial, something of what's called the trigger trial or a hybrid phase 0. Patients come in with a newly diagnosed glioblastoma. They get screened. They get given several days of this radiation sensitizer drug called niraparib. They then go on to surgery. Some of them have surgery a few hours after the last pill. Some of them have surgery 12 hours after the last pill. We need this so we can understand the dynamics there. And then when that operation is done and the tumor is removed, we ask the question, did this drug get in? You've heard about the blood-brain barrier. This is one of the biggest problems we have in our space, getting things in. Does this drug get in? If the answer is yes, then those patients go on to get the drug long-term, and we see how they do. And we'll know that whatever we're seeing in their performance is not because the drug didn't get in. If the answer is no, well, then we go on with the surgery, we finish the recovery, and we pivot to some other clinical trial. And here's a snapshot of the survival of these patients. These are called swim lanes. We call them swim lanes. So everyone begins on the left, and the longer they've been going on study and surviving, the longer that arrow is. And so this is a picture of how we look at a drug early on, and we say, is this promising or not? How does this compare to what we normally think about glioblastoma? These are unmethylated tumors. So, progression-free survival, meaning the time till first recurrence in unmethylated tumor, about 30% of patients will get past 6 months. The case you saw earlier in Tumor Board is really an exceptional case because usually they recur very fast. Drug-device combinations. This is another category of new therapies you're going to hear about. You actually are pretty familiar with it. Many of you have heard of Gliadel. Gliadel wafers are biodegradable wafers. They're FDA approved. They have a chemotherapy baked into them. You put them into the cavity. They melt into the cavity, and theoretically, they elute some of that drug into the tumor. The survival benefit has been very marginal, so it's not that commonly used, but it is approved. You also have heard about Novocure, Optune. This is the cap that many patients wear. How does it work? Well, the way it works, as best as we know, is it works in combination with the drug. All of the most promising results with Novocure have been Novocure plus Temodar. In the newly diagnosed patient, for example, that's something that's shown a survival benefit, and that was a 2015 study that was the basis for its approval. What about new drug development or drug device combinations? Well, you may have heard of sonodynamic therapy. Sonodynamic therapy is where you take a drug, you give it to a patient, and then you activate the drug with targeted ultrasound. So patients comes in the morning, they get this single shot of drug in their arm. This drug goes throughout their body and into their brain tumor, but it doesn't do anything by itself. The patients then get put in this elaborate contraption. You have to shave their head so that their head is bathed in water so the ultrasound waves can come through it. And then you target the tumor with the ultrasound. The ultrasound itself doesn't do anything to the tumor or the patient's brain. But when the drug and the tumor, or rather the drug and the ultrasound collide, that's when you see tumor cell death. And so this is us in the operating room removing that tumor and then testing it. You remove the tumor en bloc, like you see here. In this study, half the tumor is treated, half is not. So you have what's called an internal control. Because the question is always, is this going to work for everyone, or is this just something happening well in this patient? That's why it's so important to have comparisons. And so in this early study, it does seem to have a cellular effect. That doesn't mean it has a clinical effect, and that's why we're going to learn more in the ongoing trials. We talked about the blood-brain barrier. This is a perpetual problem. It's also the reason why we're all healthy in our brains, because our brain is designed to keep stuff out so that bad stuff doesn't get in. It's just that when we need to get drugs in, that's really difficult. There are multiple new strategies in development to try to get new drugs in, but to be honest, this is an under-resourced area in our field. This is such an important problem, but I can show you on a single slide almost anything that's been done to try to surpass the blood-brain barrier. You have strategies like ultrasound to disrupt the blood-brain barrier, what's called convection-enhanced delivery, which is a tube that goes in and delivers the drug directly, like in the poliovirus trials. There's what's called super selective intra-arterial infusion. I'll show you a video of this, and there's an intranasal drug that may open the blood-brain barrier. Super selective intra-arterial infusion is a strategy where you say, okay, I'm going to insert a catheter through the artery in someone's wrist, and that catheter is then going to go up their arm, through their heart, up their neck vessels, through the base of their skull, and kind of dock in the region where this glioma or brain tumor is. And when it's there, I'm going to then pulse it with one drug that transiently opens up the blood-brain barrier, and then I'm going to follow it by flooding it with another new drug, which is an experimental therapy. And the reason that this is going to be helpful is because I'm going to get a lot of drug into the target without getting a lot of drug washing out to the rest of the patient's body. Because side effects are the other major sort of contention in drug development, is how do you get drugs in safely, but also how do you combine them safely? So here's a procedure and how it actually works in a patient. And this is the first patient that was just done not too long ago. Patients under the drapes there, they're threading the needle, so to speak, through their radial artery. You see the catheter come up past the cardiac silhouette. This is the internal carotid artery, and this is all being watched in real time in three-dimensional kind of X-ray imaging. And so you can see where the tumor is getting its blood vessels from, what we call the blush, where it's being filled with blood, and then you can target it very directly with an experimental therapy and then ask the question, how much more of this drug am I getting in? And so I'll give you the results from the first patient, which is the first time anyone's heard of it as far as I know. When we gave this new drug, or not this new drug, the experimental drug, temsirolimus, to this patient, we achieved 1,000 nanomolars of the drug in the tumor. When you give this drug to them in IV formulation, you achieve 200 nanomolars. Now, the key thing is we were able to give 1/50th of the total amount of drug. So 1/50th of the dose gave us 5x drug delivery. So this is the kind of ratios that we need to have here. We need to be getting order of magnitude increases of new drug into a patient's tumor safely. So let's put all of this into practice. I will tell you, as I was sitting in tumor board, I got a text from a patient to a link to the Daily Mail article today on a new microelectric chip device that is being designed to treat brain tumor patients. So we hear about this stuff all the time. When I was putting together this talk, I was getting phone calls and emails about this article that many of you saw in the New York Post about this new drug that was in development at City of Hope. And there was a lot of buzz. And it doesn't seem incredible. It's just, where are we really at with the data on something like this? Dr. Sanai, is this something I can get on now? Is this going to help? How promising is this? Well, let's look at it, okay? The drug is called AOH 1996. It targets this mechanism called PCNA, which is a mechanism in tumor cells and normal cells that regulates cell division, okay? So targeting cell division makes a lot of sense. Tumor cells divide. I'm not going to geek out with you here, but the drug itself is really interesting because from a biochemical standpoint, it's actually 3 drugs that have been fused together, 3 really disparate molecules. And we have a medicinal chemistry group in our team at the Ivy Center, and it's almost impossible for us to even understand how these things stay together, but that's for another lecture. The point is, what do we know about this drug? Well, here's what we know about this drug that was in the New York Post. Number 1, you give it to normal cells in a dish that are dividing, and it'll hit the target and suppress the target's function. Good. Number 2, you give it to non-brain tumor cancer cells, so other types of cancer cells, in a dish, and it will suppress their growth, okay? Doesn't eradicate their growth, but it'll suppress it compared to other things, including some other older chemotherapies. Good. Next, you give it to animals that have non-brain tumor cancers, and it'll slow the growth of those non-brain tumor cancers. Not dramatically, though, okay? I'm showing you on the right in a tumor called neuroblastoma. The circled line is the drug itself, which is orange. It's kind of off to the left. It's only when it gets combined with something else that it seems to have more of an effect. So, this is evidence of some impact, but this is not, curative for any means, okay? That's all we know about this drug in the New York Post that I think some of us are probably aware of, and many of us as physicians and scientists have been asked about. Here's what we don't know about the drug. Can it get into the brain or a brain tumor? Don't know. Does it have any effect on glioblastoma cells in a dish? Unknown. Does it have any effect on glioblastomas in animals? Unknown. Is it safe to give to patients? Unknown. Is it effective in brain tumor patients? Obviously unknown. So where are we with this drug? And this is where you all can go to clinicaltrials.gov, which I hope you're familiar with as a repository for clinical trials, and you'll see that there is a single trial for this drug. It just started, and it's what's called a phase 1 trial. Remember, phases of trials. Phase 1, just trying to find a safe dose, just giving it to patients to find a safe dose. Phase 2, I know what the safe dose is. Is there any reason to think this could be effective? Phase 2. Phase 3, I think it might be effective, but the only way to find out is if I randomize patients. Some patients get placebo, some patients get the drug. That's phase 3. So this is phase 1 in solid tumors, meaning not just glioblastoma patients, any kind of patient that has a tumor that's solid, meaning not in their blood, that it doesn't have any other options. And the concept of a phase 1 is how much of this drug can we get before the patients get too ill, and then we can't give any more, and that's the dosing of the drug. So even though you're going to be encountered and bombarded with interesting tidbits of information and potentially promising ones, you're going to have to develop some of this toolkit in order to sort through, fact versus fiction and what it means for you directly, your loved ones. Here's what I'll leave you with, and I think many of you know this. Find a doctor that you can trust. As Dr. Berger mentioned earlier, these physicians are making decisions on your behalf. When I'm in the operating room and I'm operating, there's already an understanding I have with my patient of how far I'm going to push it, what the goals of surgery are, what the risks are for him or her, what they're prepared to undergo afterwards, and what they're not prepared to undergo afterwards. So find a doctor you can trust because they are going to be your representative even in times when you're not able to make those decisions. Second, don't put too much pressure on yourself. There are no secretly successful therapies. I promise you that this community will know instantly when there's something that can have an effect and is durable. It's not going to be something that you have to uncover on your own. It doesn't mean you shouldn't do your research. The newer the therapy, the more unknowns are going to be. Simple as that. That is the trade-off. When you go on new experimental therapies, your physician and that team has as many questions as you do. So that's important to understand because those questions equal to risk. Trust the data more than the narrative. Narratives can always sound good. The data is what matters. What evidence is there that this has worked in other patients? And that evidence is always difficult to come by because there can be caveats, but what has few caveats are randomized studies. And that's really the gold standard in our field of clinical research. When patients go to a phase 3 trial, and that trial is positive, then we know with a high degree of certainty that this drug or therapy is going to help you. So with that, again, really a privilege and honor to be here. Thank you again for your time and attention.
Thank you so much, Dr. Sanai. We will take a few questions. Just remember to submit it on the Whova app by clicking session Q&A or raising your hand, and a volunteer will come over to you with the mic. So we actually have a question on that last slide that you shared. So the very first point, which is such an important one, you shared, find a doctor you trust. Can you share any advice on how to do that? What are questions patients should be asking? What should they be looking for to find that doctor that they feel like they do trust and will listen to them?
Yeah, I think you've already heard some of the logical things to do here in terms of people that have experience. That probably filters out a certain proportion of doctors you might encounter. But then beyond that, it's diminishing returns, okay? I operate on 350 brain tumor patients a year. Somebody else may operate on 300 brain tumor patients a year. That differential isn't really relevant. So having somebody that specializes in what you do is important. But then beyond that, I think it comes down to the relationship and to your confidence in that individual. This is a partnership. These patients, doctors, surgeons, this is a primary care relationship. So you have to feel comfortable with your team's approach, your doctor's style. You have to be comfortable with where they are. If they're in Timbuktu and you live on the other side of the world, maybe that doesn't work for you. So I think you have to be very practical here.
Thank you so much for that advice. Another question we have is talking about research and collaboration. Can you talk a little bit about the collaboration between researchers when it comes to improving treatments? And do you have any insight on how this collaboration can be improved so that we can continue to see these great advancements?
I think, there's no lack of effort for collaboration in science. Researchers inherently are collaborative people. We all want to work together, and we're all stimulated by each other's ideas. But the lubricant for that collaboration is really funding. It's funding and lack of bureaucracy. And those two things really come hand in hand. You can flood a project with money, but if it's paralyzed by monolithic organizations with 30 levels of red tape, it's probably not going to reach where it needs to reach. And the converse is true as well. You could have a very nimble organization, but if it's underfunded, it's going to have lesser impact. So I think it comes down to that as much as anything else.
Thank you so much for that insight. Another question we have is talking about sonodynamic therapy. Can you talk a little bit more about where this is readily available? Is it readily available at many treatment centers, only a few, and how can patients find where to access that?
So, I should preface, as I showed in my disclosure slide, I have no relationship to any companies or drugs that we're testing at the Ivy Center, financially speaking. Sonodynamic therapy is being driven by a company called Sonalisense, and I think they have a booth out there. They have a multi-center clinical trial, and I believe it's at 4 or 5 centers. I think NYU, UCSF, MD Anderson come to mind. There's a pediatric version of it at Washington National. The Ivy Center also, of course, has sonodynamic therapy. So it is proliferating. Important to remember, it's a very intriguing concept, but still at the early stages of development.
Thank you so much, Dr. Sanai. I think we'll take one last question. You talked about clinicaltrials.gov, which you're all familiar with. Can you share some advice or maybe other resources on where patients and families can find more lay-friendly information on trials and ongoing research?
Yeah, I think most of the major cancer centers have a collection of resources. So for example, at the Ivy Center, we have a collection of websites, nonprofits, et cetera, that we've vetted that we think are reliable, unfiltered information. ABTA provides a similar service. So I think your local center should have that. And then, everyone has different levels of sort of facility with this information. So I've seen patients, families, caregivers at the same research meetings I'm at, and I think there's nothing wrong with that as well.
Thank you so much, Dr. Sanai. That's all the time that we have for questions. I'll go ahead and turn it back to Diane.